Welcome back. We're ready for A&P2, Chapter 26, the urinary system. The urinary system is there for primarily that number one job, the excretion of nitrogen wastes. Now, you'll remember I've spoken before about nitrogen being a very energy-rich and very reactive element. So trinitrotoluene may not sound familiar, but TNT, dynamite, may in fact ring a bell. This is a nitrogenous substance which when activated properly produces tremendous release of energy, and it's the same in metabolic reactions. It's so reactive that some of its forms are very hazardous to cellular life and must be eliminated. I'll explain how that affects the human system as we get along in the presentation. But because we have a urinary system that's eliminating nitrogen in the form of urea in a liquid state, the urinary system also has far-reaching effects. Water balance, blood volume, and pressure. That means the concentrations of all the solutes and the pH of the blood. In addition, the urinary system is the site of the production of EPO, which regulates the production of red blood cells, and also the synthesis of vitamin D, which regulates calcium levels. Nitrogenous wastes in our body result from the digestion of proteins. To some extent, nucleic acids as well, but mostly proteins. There are so many more proteins, and we use them for so many things. All the amino acids must be present for us to synthesize our proteins, but we can also use proteins for energy. And when we do, we break down amino groups. We break them off the amino acid and release amino groups in water, which forms ammonia. Ammonia is deadly toxic to cells. Now, we do a trick in the body by converting ammonia to urea. We take a carbon dioxide and stick two ammonias on it. So we concentrate the ammonia and also take it down several ticks in terms of its toxicity. We are not the only way of dealing with ammonia. Fish excrete it directly. Birds and reptiles make it in such a concentrated form and dehydrate to the extent that basically crystals of uric acid precipitate. That's that grainy feel. But our system is a completely liquid system, urine. In a healthy person, again, there are no solids or crystals in our excrement. The kidneys, ureters, urinary bladder, and urethra form the entire system. What we're going to do is take blood out of the main circulation of the body, produce urine, removing the toxic component, and return the cleaned blood to the circulation. It is not an absolute removal. So if you imagine this being the tolerable level for urea, and this being the level that the kidneys can maintain in the body, we're basically trying always to eliminate enough urea that that safety margin is maintained. The kidneys are mounted in the retroperitoneal space, high in the abdominal cavity. They are toward the back cavity and behind the peritoneum. Their connection to the descending aorta is short and a high diameter, so the blood pressure pushing blood into the kidneys is very high. An estimate of the blood diversion is about 30% of the flow down the descending aorta will make the trip into the kidneys. And here is what it sees. We have first the outer fibrous capsule of the kidney that contains the entire organ. Right inside that is a clear area, and you will notice, excuse me a minute, I don't have my pointer. I don't have my pointer activated. I'm going to pause like a rookie. And activate my pointer so I can be more clear about where I'm talking. There we go. You can see it now. Here, the fibrous capsule on the outside. Right inside that, this clear perimeter is called the cortex, all the way around the kidney in 3 dimensions. Then we have this purple swath right here. Together, these purple triangles constitute what's called the medulla. But they are individual triangles that are actually shaped more like funnels, with a broad circular top and a very narrow delivery point. These are called pyramids, and as I said, together they constitute the medulla. At the tip of the pyramid, a region called the papilla is the delivery point for the urine into these tubes. These were called calyx tubes. From all regions of the kidney, they converge here on the delivery point to the ureter. This is called the renal pelvis. So looking at the kidney, you notice that the kidney itself is rather nondescript, but you can clearly see the ureter, the pelvis, and the branching calyx. They are each hooked to medulla, those funnel-shaped pyramids that together form the medulla in the kidney. The kidney lobes are basically defined by those medulla, and then the cortex surrounds it all. The anatomy of the kidney shows the delivery of the blood. If you follow the renal artery, a branching network. It delivers blood by branching, basically taking advantage of these spaces between the pyramids to deliver blood to this entire perimeter. If you blow this wedge up over here, you'll notice that the cortex is where the beginning of the functional unit is. It is called a nephron. The nephron is the unit that actually filters the blood and makes the urine. These glomerulus heads right here, which I'll show you in a moment, are connected to a series of tubules that dip into the medulla and return to the cortex, attaching to collecting ducts that will deliver the urine down. Let's take a look at this a little closer. Here we have a fairly accurate representation of the way these units called the nephron are grouped together. They are packed around these delivery arteries with high blood pressure, delivering the blood initially to the globe that we call the glomerulus. Over here to the right, we can see that an individual nephron is drawn. Here is the delivery of blood to the glomerulus, and the glomerulus is attached to this convoluted tubule called the proximal tubule, the nephron loop, which dips down and returns, this convoluted tubule called the distal convoluted tubule, and finally the collecting duct. Different chemical processes are conducted in each section. In addition, the blood that flows out, that has been partially cleaned of the urea, then forms a network of capillaries which surround these tubes and allow for constant exchange, going both directions. We have the ability to reclaim important and valuable substances from the filtrate and put them back into the blood. That'd be called reabsorption. And we also have the ability to grab certain wastes and transport them into the filtrate. Let's start the process with the glomerulus, which is where the first step of urine formation occurs. The glomerulus is an outer capsule that contains the blood flow through the capsule and back out. Now this knot, this net-like, or I don't know how to describe this. It kind of reminds me of a ribbon, the way it is convoluted with its various branches and divisions. This glomerulus capillary network is unique in that it actually has physical holes inside those squiggly regions. The purpose of the hole is to separate small molecules from the blood. And anything that's smaller than the hole will flow from inside the capillary out into this space. The first operation is a filtering operation. You're basically like filter paper. If you're smaller than the hole, you go through. Before we leave the glomerulus and this initial step, I want to point out this group of cells right here. This is a group of cells called the juxtaglomerular apparatus. Juxtaglomerulus just means, glomerular just means right next to the glomerulus. And over here you'll notice the proximity to the distal convoluted tubule. So these nephrons are going to be drawn in kind of extended form so you can see every part, but in fact examining them is not part of their function. These tubes are packed together and wind around each other so that they can efficiently produce movement between the blood and the urine that's developing, and between the urine and the blood. So the anatomy path is from the glomerulus to the proximal tubule. This is called a convoluted tubule, and as soon as it straightens out, it becomes what we call the nephron loop, which always dips downward, turns the corner, and returns back. This is the distal convoluted tubule and the connection to the collecting duct. Notice that the blood flows in this direction through this net-like reticulate formation. These are called peritubular capillaries, just means around the tubule, and then eventually the blood returns to the venous network for exit from the kidney. This anatomy is going to divide the process of forming urine into distinct steps. We do notice, however, there's a little bit of difference in the anatomy of these nephrons. If you are mounting the glomerulus high up in the cortex toward the capsule, then the nephron loop tends to be shorter and only dip a small amount right here into the medulla. This boundary here is a separation of chemical steps from cortex to medulla. If the glomerulus is deeper down, you'll notice there's a kind of a difference in the way the convoluted tubule is formed, but then the loop dips way down into the medulla and back out before the distal tubule and collecting duct are encountered. The circulation through the cortical nephron and also then through the juxtamedullary nephron are somewhat different because of their different placement. Let's form some urine. There are 3 steps. The first step occurs only in the glomerulus. I've described it as pushing small molecules out through small holes. So that means what? Water, but also sodium, potassium, calcium, magnesium, even glucose is small enough to get through these holes. Notably, urea also goes through. This first step, filtration, is completely based on those small holes and the size of the molecules. I should mention that here, as the blood flows through, large molecules that are abundant inside the blood cells and inside the plasma, albumins and globulins, are not filtered out. They're too big. Also, all the blood cells are kept in the blood and flow out. As we move down through the nephron in all of its parts, and those are closely associated with the peritubular capillaries, 2 processes are constantly exchanging materials from urine to blood or blood to urine. The first thing that happens is constant. I mentioned that sodium and potassium and glucose are all filtered out. We want to grab those back. So that would be called reabsorption if we're moving from the urine back to the blood. However, there are some waste products in the blood, and there are conditions in the body of homeostasis that have to be maintained, and there are mechanisms to move some wastes from the blood back to the urine for removal, for excretion. That would be called secretion if we're going toward the blood. Let's take a look at this process, filtration. Blood flows through those glomerular capillaries, and you can see the yellow filtrate surrounding that material. The high blood pressure just keeps pushing, and water, urea, and all those valuable small molecules move out. Large molecules like proteins and blood cells stay in the blood. So the blood flows out of the glomerulus through the efferent arteriole. The filtrate is pushed by that pressure down through the proximal tubule and into the rest of the nephron. Reabsorption. At first it's constant. We want to actively reclaim those valuable things that we have obtained in our diet, like sodium, potassium, and chloride. Nutrients like glucose are also completely reclaimed. There are carriers, so facilitated diffusion means that there is no requirement for ATP to get those back in blood. Now, water and its balance is controlled by osmosis, and we're going to say more about that. But basically, the cell can't grab water molecules and move them. They rely on transporting ions and molecules, changing the solute concentration in order to get water to move in the right direction. We'll say more about that in a moment. Secretion. Secretion is moved from the blood to the urine. Hydrogen ion and various waste products are basically moved to the urine and excreted from the body. This is a more variable process. So, for example, you stop and you get one of those 44-ounce big gulps, and you put 44 ounces of pH 3 in your body. The pH of the blood is pushed in the negative direction. One of the things the kidneys can do is move hydrogen ion from the blood directly into the urine. It's why the urine is one of our more variable fluids, is this situational transport that we call secretion, moving things into the urine stream. Also, the kidneys often act to remove drugs from the bloodstream. So issues of medication, issues of kidney failure often depend on the ability of the kidneys to remove these foreign molecules. Here we see a very extended drawing of the nephron, spreading it out so you can get an idea of what happens in the different sections. You'll notice that filtration occurring here and that constant reabsorption that occurs here is part of the proximal tubule, but as it descends down here, a couple of things happen. Water is removed from the stream. It's a pretty dilute up here, but as you remove water, it gets more and more concentrated as we move down to the bottom of the nephron loop. On the ascending loop, we transport out specifically sodium ions and then up here co-transport of sodium and chloride ions, and as we remove the solute, the filtrate becomes less dense again as we enter this distal convoluted tubule. This distal tubule is going to be a place where the conditions of the body largely determine what is moved. And so we have the ability for reabsorption and secretion in that distal tubule. And we also here then, in the collecting duct, we have an ability to basically affect the water balance. What does this shading mean from nearly white down through pink to deep red? What we're doing down here where it's deeper red is we're producing an osmotic zone with very high concentrations of solute. Now remember, a very concentrated solution, it doesn't make any difference what is dissolved in it. It produces the same kind of osmotic pressure. The higher the concentration of solute, the lower concentration of water. So if you have a dilute solution going through here and down through the collecting duct, if there are open channels, that water will move right out, conserving water. If those channels are closed, then the dilute urine will pass out through the collecting duct and be emitted as dilute urine. Reclaiming water depends on this process, where the solute concentration causes the diffusion of more water from the loop of Henle and basically would reclaim it and retain it in the body. Notice that it's not just sodium and chloride, but down here, one of the molecules you have available in great abundance is urea. And so it can be used to increase the osmotic gradient. We can adjust this and situationally, so whether you're overhydrated or close to dehydration, we can control the dilution or the concentration of the urine through controlling the passage of water out of this filtrate. Hormones like ADH, antidiuretic hormone, produce this effect. Here is a comparison on the left. You'll notice a couple of things, and I like this drawing not just for the ADH, but watch this. Here's the filtrate. See how concentrated the solute is? As you pump the water out, look, it gets more and more concentrated. Turn the corner and the pressure pushes it back upward, and now you're pumping out sodium chloride molecules and then sodium and chloride ions and diluting it back to kind of its initial state. You've reclaimed most of what's useful, water and ions, and the dilute urine comes here. If there is no ADH, antidiuretic hormone, in the system, then there's no passage of water out of the collecting duct, and that dilute urine is passed out. So this is the condition that happens. You're sitting and drinking and you have 5 beers. You're basically going to be going to the bathroom to release high volumes of low concentrated urine. But what happens when you get lower and lower and lower in your hydration? And you're close to that border of dehydration. Here's the same process of concentrating and diluting. But then if water is close, marginal in its availability, we have ADH released. It's actually released from the brain region. The pituitary gland releases antidiuretic hormone, and it affects the distal tubule and the collecting duct, allowing water to respond to osmosis. Now that water is drawn out, conserving the water. Again, your body is dehydrated, so the kidney is conserving water, and as it passes down through the medulla and the collecting duct, it is concentrated to higher and higher degrees. A small volume is emitted, but it's very, very concentrated. The values of these different ions, metabolites, and nitrogenous waste are shown here. The normal homeostatic range that we encounter is shown in plasma, and the urine shows how the urinary system, especially the kidney, can form urine. So let's start up here with the solutes. Sodium, potassium, chloride, and bicarbonate are present in our plasma at all time. Look at these relatively narrow ranges. That means they're under homeostatic control. So we basically see 3.5 to 5 milligrams per deciliter. I'm sorry, milliequivalents per deciliter. But over here in the urine, the reason we can maintain this is that we can concentrate potassium just a small amount, or we can dump excess potassium. In each case, these ranges begin at the around the high level and go well above the maximum level, with the exception of bicarbonate. Now, the reason bicarbonate is important here is that you have a different way of getting rid of bicarbonate. Your respiratory system is constantly changing bicarbonate back to CO2 and breathing it out. But these three, sodium chloride and potassium, are all dependent on diet. So if you have a lot of extra sodium, let's say you eat a lot of salt, so your sodium and chloride concentrations go way up. Look at the level you can excrete in your urine. Now, metabolites and nutrients are valuable. And what's distinctive about this category in milligrams per deciliter is the very, very small amounts of these nutrients that are passed out in the urine. No detectable protein, almost indetectable glucose. Lipids and amino acids equally low compared to the amounts that we find in the blood. Now the ranges here are much more variable. Glucose 70 to 110, and it's just an indicator of the change in concentration that's related to the absorptive state right when we've eaten and directly after we eat, and then the lower concentrations that occur between meals. But nitrogenous wastes, these levels in the blood plasma are ranges considered acceptable, and look at the efficiency of the kidney in eliminating urea, up to 1,800 milligrams per deciliter, or 1.8 grams of urea per deciliter per 100 mils. Likewise, creatinine, uric acid, and ammonia, those which are present in much lower trace amounts, can be concentrated in urine to rid the body of excess nitrogen. We do see an effect on blood pressure and volume. Here's a normal blood pressure and volume beginning, and a situation where blood pressure and volume are decreasing. The kidneys respond with a dual action, based on the reduced renal blood flow and the reduced oxygen content of that blood. Renin is released by the kidneys, and it produces what I call the renin cascade: angiotensinogen, angiotensin 1, angiotensin 2, and aldosterone are the sequential enzymes that are produced, and their main effect is multiple. It increases sodium retention, which means the water stays in the body, so this is working on water retention. It causes the secretion of ADH, which also works on fluid retention, concentrating the urine as much as possible. But the third effect is probably the most immediate. You get thirsty, and so you drink a couple liters of water. That immediately increases the blood volume, and pressure is restored. This renin cascade is the shortest term response to lowered blood pressure. This one in the center, erythropoietin, is released, and that affects the red bone marrow to produce more red blood cells. In a matter of, let's say, days, the hematocrit increases as more red blood cells are produced, and that swells the blood volume back to a stable and acceptable level. The juxtaglomerular apparatus that we spoke of, located kind of right in here, is the source of both erythropoietin and renin, and what a perfect place to sense blood quality. This is where the blood is flowing by, and it is the direct organ which is regulating water release. So pressure and volume relationships make a lot of sense. Erythropoietin is a peptide hormone, and its effect is on the red blood cells produced by the red bone marrow. It basically dials it up a couple of notches. Now, atrial natriuretic peptide deserves some mention. Those hormones that we mentioned from renin, angiotensinogen, angiotensin 1, angiotensin 2, aldosterone, all have that effect toward retention of water and maintenance of blood pressure. But what happens when blood pressure goes the other way? Atrial natriuretic peptide is released by the atrium of the heart, and it basically counters every one of those effects. Your thirst goes away, so you don't drink. It basically stops the release of antidiuretic hormone, and it basically closes those water reabsorption transport channels in the distal convoluted tubule and collecting duct so that water flows out through the urine and high volumes of dilute urine are released, bringing blood pressure and volume back down. Water and salt balance, the solute concentrations, are regulated as well by the kidneys. The cell can move solutes. It can't actually move water. So we have a saying to help us remember what osmosis does. Pump the salt and the water follows. Water follows salt. And so let me imagine my body here is a membrane, and I have an isotonic concentration on each side, equal concentration of salt molecules. So that means equal concentration of osmosis. So even though water's going back and forth, it's at an equal isotonic level. Now what happens if I activate pumps to pump solute from this side, my left side, to my right side? The concentration of water as salt is pumped out goes up. The concentration of water as salt is pumped in goes down. So the water concentration is going like this. Osmosis is the diffusion of water across a semi-permeable membrane. So if you pump the salt in this direction, the water is going to follow. And this mechanism is how we control the water balance. Very important in our cells to maintain proper osmotic relationship, solute concentration, size, and shape. The kidney also helps maintain pH, and this shows that above that CO2 and water system, we're always making CO2 from respiration. It is converted into carbonic acid there in the center and to bicarbonate and hydrogen ion on the left side. So what does that mean? When we eat, we often eat acid foods, and we often, as we exercise, generate excess CO2. Now, some of that we're getting rid of by our breathing. As we breathe the CO2 out, water is left behind, and excessive hydrogen ion is moderated. However, sometimes it still needs a little extra help, and what happens is secretion as the kidney helps maintain pH. The hydrogen ion is pumped into the bloodstream, where it combines with ammonia to form ammonium, and out it goes. The bicarbonate is pumped in the opposite direction from the urine back into the blood capillary flow, and pH is moderated. So in thinking about pH control, always think about respiratory and urinary systems together. They help maintain it by secretion in the kidneys when hydrogen ion is excessive, and by the constant regulation of our respiratory center by breathing the CO2 out. So to some extent, you are affecting pH just by breathing faster. So I like this diagram. When we do a lot of introduction, when we do a lot of analysis, I like a diagram that summarizes everything we've said to some extent on one diagram, and here it is. An extended or pulled out version, a drawing of the nephron, showing the location of the glomerulus and filtration. And then the counterbalancing effects of reabsorption and secretion. Are they constant or are they variable in the other portions of the nephron? So if you see an arrow colored blue, it's water movement. If you see an arrow colored yellow, we're transporting solutes. If the border of that arrow is solid, it's constant. It's going on as long as the material flows. And if you see it with a dotted line, that means it's variable. It happens only when it's needed. So in the proximal tubule, you see constant transport of water and solutes out into the surrounding regions. In the loop of Henle, the nephron loop, you'll see a constant transport of water out in the descending limb and a constant transport of solutes in the ascending limb. So what's left behind is the nitrogenous waste and other waste materials. As we get into the renal tubule called the distal convoluted tubule, shown right here, you'll notice that those borders are now dashed lines, meaning that they are variable for reabsorption or secretion. When needed, solutes are transported into the tubule, or when needed, transported out. Notice that there are arrows that are pointing in both directions for reabsorption. That's very situational. What is in the urine, what has been filtered out, based on the peculiarities of your day, your diet, what you have taken in and what wastes are being produced. Here you'll notice water out is an option when you're trying to conserve water, and as it gets into the collecting duct and probably affected by the presence or absence of ADH or the renin cascade, we basically have the ability to transport solutes in either direction, but also to dehydrate that dilute urine into concentrated urine when it is important to retain water. Down here at the bottom of the collecting duct, that's where you have the papilla of the medulla, and we're delivering that water into the tube network that we call calyces, the calyx, leading to the renal pelvis. Urine production is over when you collect the urine here in the renal pelvis. So it collects there, and you can kind of see the outline of this calyx arrangement, branching tubes from the papilla of each medulla, bringing its contribution to the droplets of urine that are collecting here at the top of the ureter. In order to get this pyelogram, what you do is inject an opaque dye in through the bladder and the ureters to illustrate what those tubules contain. The ureters are retroperitoneal muscular tubes connecting the kidney with the bladder. They are, as it says, muscular tubes. The openings on the bladder are slit-like and have the ability to contract slightly to restrict the backflow of urine, and the muscles in the walls of the ureter produce peristalsis, a rippling wave of muscular contraction. So when a couple of droplets form there at the top of the ureter, zip, zip, they are delivered immediately to the bladder. Contractions twice a minute are in the background and happening constantly throughout our day and night. The bladder's hollow and muscular and stores temporarily. A liter is a good estimate of its volume, and its lining was a special tissue. If you recall back to tissue, transitional epithelium is affected by how much urine is being stored at a particular time. This triangular region right here is called the trigone. Here are the two ureteral openings in a muscular triangle that basically places the fluid in this bladder, and it can fill up from bottom to top, acts as a funnel when we open the urethral sphincters and allow the passage or micturition of urine. Here is one sphincter, the internal sphincter. Here is the external sphincter. This is obviously a drawing from a man because we see the prostate gland surrounding the urethra on the way below the bladder. We do see a distinct effect with respect to pathology based on gender. The urethra descends from the male bladder through the prostate, where it is joined with the genital tract, and then it is long, extending out of the body to an end that is located far away from the anus, but also it is in an area which is relatively light, relatively dry, and kept relatively clean. Light, dry, relatively cool as well, since it is removed from the internal part of the body. That means that urine is flushing down here. Urine is a mild antiseptic, so the difficulty of getting bacteria, this is the closest reliable and repeated source of bacteria, all the way around past this barrier to the tip, and then by natural processes all the way up to the bladder. It's very difficult in the male. So males suffer very rarely from urinary bladder infections. Compare that to the female, where the urethra is relatively short. Look at this. And where it opens is in a fold between the labia. That means that the opening is moist and dark and warm compared to the surface of the body. It also is a short distance from the rectal opening and the source of bacteria. So females are much more vulnerable to urinary bladder infections because of this anatomy. Now, release of urine, or micturition, is keyed by stretch receptors in the bladder, letting you know when your bladder is half full or more, and voluntary relaxation of those sphincters allows the voiding of the bladder, typically by gravity. Urinary problems, inability to hold urine, or incontinence can result from a number of different problems. Sphincter muscles losing tone will release urine, just like voluntary relaxation. If the nervous system is damaged and the control of that those two sphincters is achieved by a stroke, damage to the cerebral cortex, or hypothalamus, or Alzheimer's disease are all known to affect the control of those sphincters. In males, urinary retention, especially in old age, is affected by this prostate. As age progresses and hormones change, the prostate tends to swell, and it basically swells on the outside, but it also puts pressure on this urethra. So that would first be seen as a slowing of the release of urine, but it would, in the initial stages, allow the bladder to be completely voided. As it progresses, you would be able to put enough pressure on this, so now when this pressure closes the urethra, there might still be 100 or 200 or 300 mils of urine left in the bladder. That leads to conditions and symptoms like weak stream, the urge to frequently avoid the bladder, and the inability to sleep through the night. In extreme situations, the bladder, prostate swelling can completely close that passageway, and the bladder will swell up to painful proportions and require medical assistance in order to drain it through a penile catheter. Now, the rest of the slides on this PowerPoint basically reinforce and amplify some of the conditions and some of the mechanisms present in urine formation. But I'm going to stop this recorded presentation at the basic end of this slide right here. So thank you very much. That is the urinary system. Where are you? And I will stop recording.